Our last topic for the macromolecules are going to be the nucleic acids. And with this video, this one's going to be not in a whole lot of detail on nucleic acids, because we're going to talk about a lot more detail with nucleic acids when we get to the chapter on DNA and RNA within the textbook. Okay? What I want you to focus on right now with nucleic acids, we've got two types, all right, or two flavors. We've got DNA, which is deoxyribonucleic acid, which is a mouthful, which is why DNA is fine, okay? And RNA, which is ribonucleic acid. And I'm going to draw each one up here, all right? And in the context of our nucleic acids, okay, the DNA and the RNA, these are our polymers, okay? Our monomers are going to be the nucleotides, all right? And I'm going to draw up here first a nucleotide for DNA, all right? And associated with that nucleotide, we're going to have three components, okay? We're going to have a phosphate group. We are going to have a sugar, and we are going to have a nitrogenous base. Okay? So it's easiest, remember, I draw my phosphates just as the P. Okay? So there's our phosphate. Our sugar is going to be a pentose sugar. Oops. Pentose. Pentose meaning 5. So we've got 1, 2, 3, 4, 5 carbons associated with it. And I should have actually counted the other way, because the way they're numbered, this is the number 1 carbon, this is number 2, this is number 3, this is number 4, this is carbon number 5. Okay? The pentose sugar is going to be what determines whether we've got DNA or whether we've got RNA. Okay? What the difference is going to become apparent when I draw the ribose next to it, but essentially it's going to get to our functional groups. What makes the difference between DNA and RNA is going to be what's attached to that second carbon, okay? So our pentose in this case is going to be deoxyribose, all right? So we've got our, in green, our phosphate group. And again, I'm not concerned that you know and draw the structure of a phosphate group, all right? And then attached to the sugar is going to be our nitrogenous base. And with deoxyribose, we've got 4 nitrogenous bases. Okay, we've got adenine, we've got thymine, we've got cytosine, and we've got guanine. Okay, and they're abbreviated A, T, C, and G. So I just put an adenine in here, okay? just for example, this could also be a T, a C, or a G. All right? And in terms of the nucleic acids, this now is our monomer. Okay? You've got three components that make up the monomer. You've got the phosphate, the sugar, the nitrogenous base. Okay? And this is going to be a monomer for DNA. All right? Our RNA structurally is going to be very, very similar. However, there are some differences that are going to allow the cell to distinguish between RNA and DNA. And the way that we're going to distinguish that is going to be with the sugar. All right? I've got a hydroxyl group here. I'm going to put a hydroxyl group here. If you notice, I've got a hydrogen here. For ribonucleic acid, our functional group is going to change. Okay? Our pentose sugar in RNA is going to be ribose. And the ribose has the hydroxyl group there, the OH. All right? Now attached to the sugar, there's still going to be a nitrogenous base. And I drew an A here as well, okay? RNA also has 4 nitrogenous bases. Those nitrogenous bases now still going to be adenine, cytosine, guanine. But what RNA has, and this is the other thing that's going to make it different from DNA, is it has a uracil instead of a thymine. Okay? So the 4 bases associated with RNA are going to be A, U, C, and G. So the nitrogenous bases differ. Our functional group on the sugar differs. So you have a different sugar between RNA and DNA. So what I've drawn here is going to be a monomer of ribonucleic acid. All right? What you're going to do in terms of linking, okay, to make the polymers. DNA, you would link nucleotides of DNA together to make DNA. You would link nucleotides of RNA together to make RNA. So essentially what you have when you're putting together DNA, let's do pink and green, pink and green, and then we've got blue with our bases. Where those are going to be linked together are going to be at the sugars, okay? And what that's going to generate with DNA is a sugar phosphate backbone. And what you're going to have on the other side over here, because DNA is a double helix, meaning you have 2 strands put together. All right, and these sugars are going to be linked. And I'm drawing this very abstractly right now because I'm not concerned right now that you understand how they're put together. We're going to go over how they're put together in more detail later this semester. Just notice the sugar groups that are going to be linked together, okay? The outside of the DNA double helix is going to be the sugar phosphate backbone. The interior structure of the helix, because if you think of this like a spiral staircase, The sugar phosphate backbone is kind of like the handrails. The bases, nitrogenous bases, are like the stairs of our spiral staircase. There's going to be hydrogen bonding that holds the stairs or the bases together. Okay? So this is our hydrogen bond. And if you notice, I kind of drew 2 dotted lines between adenine and thymine, 3 hydrogen bonds between guanine and cytosine. That's not a mistake, okay? Adenine and thymine have 2 areas where they hydrogen bond. Cytosine and guanine have 3 hydrogen bonds, all right? And we'll talk a little bit more in detail about that when we get to the nucleic acids. But if you notice, I paired these up. A is always going to pair with T in DNA. G is always going to pair, I drew another G, that was a mistake, with C, okay? So if I give you a linear sequence of DNA that goes A, T, G, C, G, C, T, you can tell me what the complementary or the other side of the DNA helix is based on this sequence. A always pairs with T, A always pairs with T, G and C, G and C, G and C, A. Okay? So in terms of DNA, you're always going to have 2 strands of bases that are going to represent the strands of DNA. Okay? And in terms of the overall structure, the ATG, that's going to be the nucleotides linked together. So here I've got the A. The next nucleotide that would be added after this one would be a T. Next one after that would be a G. Okay? So that's going to be DNA. All right? RNA, I said, was a little bit different. RNA, if I put up a sequence, you should be able to tell me whether a sequence is RNA or if it's DNA. And the way you can tell that difference, RNA is going to have the uracil. Okay? RNA also is going to be present as a single strand, okay? Which is different than DNA, which I said was a double helix. You've got 2 strands with DNA. RNA, you've just got a single strand. Right? Hopefully that makes sense. If you have any questions, please let me know.